I should mention here the value and importance of achieving a complete remission as a strategy right now for immune reconstitution. Okay, so this is my most important slide, and it gives you an idea about the direction that we're going. As I mentioned, we're making a lot of effort, and attention is being given to understanding the transformation events that occur when patients develop resistant disease, refractory disease, particularly to the small molecule inhibitors, as well as what are the risk features that lead to development of Richter's transformation. Second cancers are more common in patients with CLL, and we need to have an understanding of why that occurs. Is it related to immune suppression, and how do we fix it? And secondary cancers are cancers that are caused by the treatment, generally speaking, that we give for CLL, things like AML and MDS that relate to exposure to the alkylating-based agents. Minimizing that, reducing that is certainly a goal and effort. And as I mentioned, minimizing infections. In terms of the direction that we're working on going for the elderly population, again, as I mentioned, controlling the disease may be adequate, and developing strategies for those patients to have tolerable means of controlling their disease, whether that's in combinations of the small molecule inhibitors or sequencing of the small molecule inhibitors, is probably the strategy that you're going to see in terms of available clinical trials. And then again, immune restoration for those patients is critical and key. For the younger patients, conceptually I think of them in terms of those that have a mutated V gene versus those that have an unmutated V gene. For the patients with a mutated V gene, it's important, I think, for us not to dispense of chemoimmunotherapy that we know is effective for a good number of these patients, but to try to minimize the risks with it and to mitigate the problems that we see with the FCR combination in particular. Reducing the number of cycles required, and in that way potentially minimizing the associated toxicities. So we're working on clinical trials that will reduce the number of cycles and making modifications like replacing rituximab with GA101, adding the small molecule inhibitor, for example, ibrutinib, in a clinical trial strategy to improve outcomes and achieve a high rate of MRD negative remissions for the mutated cases. For the unmutated cases, my opinion is that those patients are probably best off avoiding chemotherapy as long as they can. Even with FCR, we know we can get a number of them in a good, complete remission, which is MRD negative, but they relapse. Almost all of them relapse. And so we know that chemotherapy has activity in those patients, and I think those patients probably will benefit with some time now, because things are happening so quickly and the standard of care today is going to be different. The available drugs that we have for study are different now, and working on testing and evaluating new and developing new agents, particularly for the unmutated cases, and potentially curative strategies, cellular therapies, etc., would, for me, be ideal for that population, and waiting and delaying as long as possible exposure to chemotherapy for those patients. You'll see more and more, I think, available clinical trials with regard to consolidation. So can we achieve a good remission or a good disease control state with ibrutinib, for example, and then add a new agent, a checkpoint inhibitor, monoclonal antibody, a cellular therapy, and in that way consolidate and eliminate that residual disease that is there, so that patients can get a good, durable remission and have some treatment-free period. Maintenance, I think, is also an important concept. So I'm just going to touch on this slide. All of these, most all of them were touched on in terms of strategies, combinations and sequencing of small molecules, and combinations of the small molecules with the agents on the lower half of this slide, the new drugs in development, the checkpoint monoclonal antibodies. Monoclonal antibodies, as Tom spoke about, against ROR1 and other surface molecules in CLL cells. Cellular therapies are very important, and I think will be certainly an area of clinical investigation and hopefully in advance for patients with CLL. Somebody, I don't know who it was, touched on lenalidomide, and there's a new version of lenalidomide called CC122, which is an immune modulating agent that will be in study. Vaccines are also, again, an area that we've been working on for a number of years, and that work will continue. And this slide highlights, this is pretty much my last slide, highlights the new immune modulating antibodies that are in development that we've started and others have started working with. Dr. Gribben talked about PD-1, PD-L1. We'll have clinical trials with both of those antibodies against both of those targets. CTLA-4, LAG-3 is another of the checkpoint inhibitors. CD137 is an immune modulating, is a target for immune modulating antibody, as well as KIR, which is a molecule on the surface of NK cells, and antibodies can activate those cells after binding. So my take-home thoughts are, we've heard this also today, no treatment progress possible without the courageous patient participation in our clinical trials and clinical research. And I know all of my colleagues among the CLL community are very grateful for the courageous participation that you all take in our clinical work and clinical investigation, and there would be no advance or progress possible without that. There's still a lot of work to be done. We have a lot of opportunities now, more opportunities than we ever did. I think my suggestion to you would be to be informed, as you've heard already today, and seek clinical trials and opportunities that are smart and that have some benefit to you. And as far as I'm concerned, we've never been closer to having a long-term control and potential cure of CLL. So I'm very optimistic about our future, and with that I'll close.